In this presentation, we show the results of two pre-emergent herbicide trials in plant cane in far north Queensland. These trials were part of the project called Developing an Alternative Herbicide Management Strategy to Replace PS2 Herbicides in the Wet Tropics, run by SRA weed agronomist Emily Phelos and funded by SRA and the Queensland Government. One trial was located at Mirrawinny, just north of Innisfail, and the other was at Craiglee, near Port Douglas, north of Cairns. Pre-emergent herbicides, either on their own or in a mix, were applied to plant cane at the spike to 3-leaf cane growth stage. Weed cover measured at 15 and 30 days after spraying was compared to unsprayed plots. Results from the two sites have been combined for this presentation. These herbicides were tested on both trial sites. Active ingredients are in brackets. These herbicides were tested on one trial site. Active ingredients are in brackets. Weed pressure at the Mirrawinny site was mainly guinea grass, whilst at the Craiglee site both grasses and vines were present. All the herbicides gave effective grass control over 30 days of assessment. Although there were some slight differences, all tended to give similar control of grasses, about 96% reduction in grass germination after 15 days and around 92% reduction in grass germination after 30 days, in comparison to the unsprayed plots. All the herbicides tested are grouped as one line on the chart because their effectiveness was so similar. Control of grass germination would have extended past the 30-day assessment, but in these trials weed cover was only measured out to 30 days after spraying. Here we see the typical level of control achieved 30 days after spraying, in comparison with the unsprayed plots. Looking at vine control, all the herbicides tested, except Balance by itself, gave very similar control of vine germination over the 30 days of assessment. The orange line in the chart depicts how the herbicides, excluding Balance alone, achieved around a 96% reduction in vine germination over the 30-day assessment period compared to the unsprayed plots. They are shown as a single line on the chart as on average they gave very similar control. As expected, Balance by itself was less effective than other pre-emergents in controlling convolvulus vines. This is shown by the lower blue line in the chart, with Balance by itself giving around an 87% reduction in vine germination over the 30 days. This is why Balance is generally used in a mixture, for example mixed with a metribuzin herbicide like Mentor. The length of weed control achieved by pre-emergence may be influenced by application rate, with higher application rates generally achieving longer periods of control than lower rates. Soil texture and soil chemistry. Ultraviolet light, which degrades some herbicides. Rainfall may also influence length of control, with high rainfall intensity on lighter soils reducing length of control. Herbicides like imazapic, isoxaflutol, flumioxazin are effective pre-emergents in plant cane, with similar effectiveness to the more traditional herbicides like pendimethalin, amitrin, metallochlor and atrazine. They also have the advantage of being UV stable. However, you do need to know if they have specific weaknesses. Imazapic, found in products like Flame and Spark, is poor on crowsfoot grass and calipo vine. Isoxaflutol, the active ingredient in herbicides like Balance, is poor on squareweed and convolvulus vine. Metribuzin, the active ingredient for herbicides like Mentor and Tomahawk, gives poor control of balsam pear. With some herbicides such as isoxaflutol and imazapic, crop injury is possible from root uptake on lighter soils, so read the label for specific instructions. Crop injury symptoms from these two herbicides are usually a bleaching and or yellowing of cane leaves. It may vary in severity depending on application rate, soil texture and rainfall or irrigation. For more detail on these trials, contact SRA. We would value your opinion on future weed management research. Please take 10 minutes to complete the online survey by clicking on the link in the email you received.